Hello everyone. Yes, chapter 2, class 12, biology. It comes under reproduction unit. So name of the chapter is sexual reproduction in flowering plant. So today we will start our second chapter, that is sexual reproduction in flowering plant. So the first term which I can see here, that is angiosperm. What is the meaning of angiosperm? Angiosperm, it means flowering plants. The plants which we look around us, all these plants are comes under angiosperm. So angiosperm means flowering plant. Now the second term is flower. What is flower? Flower is nothing. It is the reproductive organ of a plant. It is modified shoot. It is nothing. It is the modification of shoot in bearing. Next is, that is pre-fertilization events. So pre-fertilization event means gamete formation and transfer of gamete. So pre-fertilization of event. Several hormones and structural changes result in the development of a flower. That means development of flower, it is the result of hormonal changes. Like human being, hormones are present in plants also, like auxin, gibberellin, cytokinin. So development of flower or appearance of the flower, it is because of the hormonal change. Another term we can see here, that is inflorescence. What is the meaning of inflorescence? Inflorescence means arrangement of flower. That is inflorescence. So already we discussed about that the flower, it is a reproductive part of a plant, and the flower which bears sepal, that is outermost part, group of sepal known as calyx, petal, it is the colorful part, group of petals known as corolla, then anther, filament together makes stamen, which is a male part, and then stigma, style, ovary together makes female reproductive part, that is corolla or pistil. And the basal part of the flower, it is known as thalamus. So thalamus is the base of the flower. So this is the structure of flower. Now what is the difference between androecium and stamen? Group of stamen is known as androecium. At the same time, the term gynoecium. Group of carpel or pistil known as gynoecium. So this is about the meaning of these all terms. So next, now we can see the next part of this video, that is structure of stamen, microsporangium and pollen grain. Here I'd like to say this microsporangium, pollen grain, these things are very important because you will get the similar term for this microsporangium and for this pollen grain. First of all, what is stamen? Structure of stamen. Already we have discussed, that is stamen consists of filament and anther. But the interesting thing is the term bilobed structure. Structure of stamen, it is made up of stamen, made up of filament and anther. And what is the characteristics of anther? That is bilobed structure. Here I will show you one picture. There you can see the structure of stamen. So this is anther and this is filament or stalk. I told you bilobed. Anther, it is bilobed. So what is bilobed? One, and this is second. So this is called bilobed structure. And then the second thing is about the next term, that is diathecous. Diathecous. So what is the meaning of diathecous? Each lobe consists of two theca. That is why it is called diathecous. So a typical anther is bilobed and each lobe is diathecous. Now I'll show you the theca. Here, as I told you, this is one lobe This is another lobe. Okay. So this is about lobe. We can write here it is lobe. So bilobed. Now the next is about theca. This is one and this is another theca. So we can write here it is theca. Theca. So it is diathecous structure. Clear? Now the next, that is line of dehiscence. Line of dehiscence. So this is line of dehiscence, means this is the part, this is the area from where pollen grain disperse. After maturation of this everything, that means whole structure, when the pollen grains forms here, these pollen grains disperse through this line of dehiscence. Now, next term is, that is pollen grain and pollen sac. What is the meaning of pollen grain and pollen sac? Pollen sac, sac means we know that it is chamber. So each theca is forming sac-like structure, chambers. These chambers are known as pollen sac. And each pollen sac, they consist or they store pollen grains. So these two terms are related to each other. The chamber-like structure which stored pollen grain known as pollen sac, and pollen grains are present in the pollen sac. Now as I told you, you will get different terms for the same structure. So pollen grains, what other term we can use for this pollen grains? So we can write here it is microspore. Microspore. So pollen grains are known as also microspore. At the same time, what other term we can use for pollen sac? We can use here it is microsporangium. So I think it is clear what is microsporangium. Microspore bearing structure is called microsporangium. Pollen grain bearing structure is called pollen sac. So you can be asked by any term. That means either microspore, development of microspore, development of pollen grain, means same. So this is about this structure of stamen. I think it is clear. Two term very important. It is bilobed and diathecous. And other term for pollen grain, microspore. Other term for pollen sac, microsporangium. When we use the term for sporangium, it means it is for singular. When we are using the term sporangia, means we are talking about several, means it is a plural term. So microsporangium and microsporangia. Now the next is structure of microsporangium, means we are going to talk about the structure of pollen sac. Once again, I'd like to tell you that which structure we are going to talk about. We are going to talk about this structure, that is microstructure of microsporangia. Now these microsporangia, we can see here wall of this microsporangia. This is the wall layer of this microsporangia, and you can see here the butterfly like structure. So it is just like a butterfly. So when we make a section of this anther like this, when we cut the anther like this, we can see this butterfly like structure, which inside of this butterfly like structure you can see here pollen sac or microsporangium, and inside of the microsporangium microspore or pollen grains develop. Right. Now what is that means I want to tell you about the wall of this microsporangium. The structure of this microsporangium. That means I am going to tell you about the structure of this microsporangium, wall of this microsporangium. Yeah. So the next topic is structure of this microsporangium. Here, structure I will show you, but before that, just let me read this. The microsporangium is surrounded by 4-wall layer. Here again it is important that it is surrounded by 4-wall layer. What is the name of these layer? Epidermis, endothecium, middle layer and tapetum. These are the name of this 4 layer. The outermost layer, that is thick and it is a protective in nature. The outermost, it is protective and it is called epidermis, first layer. And it helps in the dehiscence of the anther to release the pollen grain. So this is the function. And the innermost layer, that is, it is tapetum. So tapetum is important. Why? Because it provides nourishment. Tapetum provides nourishment for the developing pollen grain. So this is important. So here it is only the explanation of this microsporangium. Now I will show you the structure of microsporangium. Then you will come to know what is microsporangium. So this is the diagram of microsporangium. In the young anther, the sporogenous tissue forms the center of each microsporangium. Means center means which area? Center means this area. This is the center. So the sporogenous tissue, when anther is very much young, means it is just newly formed anther. Pollen grains are not formed there. It is not mature anther. That means pollen grains are formed after when the anther or when the stamen attained its maturity. But before that, how pollen grains are formed? Before that means when there is only a newly anther is produced. Inside of this microsporangia, there is no pollen grain. And the tissue which is present inside of these microsporangia, it is known as sporogenous tissue. It is known as sporogenous tissue. So suppose this is sporogenous tissue. And what is how we can represent this sporogenous tissue? Likewise, it is nothing. It is only this way. No pollen grains are there. It is sporogenous tissue. And these microsporangium means these two theca and these microsporangium, it is surrounded by 4 layers. Already I told you. So one is epidermis. This is outermost protective layer. Just it is followed by endothecium. It is thick walled layer. It is just below the epidermis. It is a hygroscopic in nature, means it can retain water. Then after 2 to 3 middle layer, and then most important and interesting layer, that is tapetum. Why tapetum is important? Why I told you that it is important? Because of two functions of this tapetum. Number one function, it provides nourishment. Nourishment to the young pollen grain. And number two, that it shows ploidy. It shows ploidy. So you will get question in your board paper, that function of tapetum. In plant only, this is the only cell which shows ploidy. So two functions of this tapetum, or two important features relate with this tapetum. One is it provides nourishment to the developing pollen grain, and second is showing ploidy. What is the meaning of ploidy? Ploidy means one cell having more than one nucleus. That is called ploidy. As we are saying, haploid means one set of chromosome. Diploid means double set of chromosome. Likewise, when a cell contains more than one nucleus, that means they are showing ploidy. So this is generally we know that one cell, one nucleus. But here in tapetum layer, they are containing many nucleus. Here, many nucleus in tapetum. So it is ploidy. So ploidy means many nucleus in one cell. So this is the characteristic feature of tapetum. So once again, this is the structure of anther. You can say it is a mature anther. So first of all, we are going to see that what is the structure. It is microsporangium. It is also known as microsporangium. 4 microsporangium we can see here, or pollen sac. Now the structure of this microsporangium, wall of this microsporangium. So the outermost wall is known as epidermis, followed by endothecium, thicker wall, then 2 to 3 middle layer, and then innermost wall, that is tapetum. So these are the name of this wall. And what I told you, that is about when the anther is young, the pollen grains are not formed. At that time, it is this pollen sac is filled with same type of homogeneous type of tissue, that is called sporogenous tissue. That is called sporogenous tissue. Okay, right. Now I will show you the diagram which is given in your book. The same diagram. It is asked in your CBSE exam that draw the tears of anther. So don't be confused. This is the butterfly-like structure, and you can draw a single portion, and you have to underline or write or label the diagram. That is epidermis, endothecium, middle layer, tapetum. Okay. Now the next topic is microsporogenesis. Here already what we were talking right now, that is about the structure of microsporangium. Now microsporogenesis. What is the meaning of microsporogenesis? Microsporogenesis means pollen grain or microspore formation process. The process of formation of microspore. Genesis means formation. Genesis means formation. So the process of formation of microspore is called microsporogenesis. Now, how it occurs in anther? We want to know this. As development occurs in the anther, the sporogenous tissue undergo meiosis to form microspore tetrad. Here I want to show you, first thing is sporogenous tissue. I told you that the young anther, there is no pollen grain, all the tissues are same and they are known as sporogenous tissue. Now this sporogenous tissue, here I can show you where which one is a sporogenous. This is sporogenous tissue. Now these sporogenous tissue, that means tissue means a group of cells. So each cell can undergo meiosis process, and these cells are known as microspore mother cell. Another term what we can use for this, in chapter 1 I told you, that is meiocyte. Meiocyte means the cell which undergoes meiosis. So each cell known as microspore mother cell. So out of these all cells, some of the cell or most of the cell, they undergo meiosis. And after meiosis, we know there will be production of 4 haploid cells. And these 4 haploid cells, it is nothing. It is, first of all, that means at the initial stage, they comes under, that means they are together in one particular structure, 4 haploid cell present. So it is, this structure is known as pollen tetrad. This is known as pollen tetrad. Okay, it is written here. No need to write again. This is known as pollen tetrad. So once again, young anther, when there is no pollen grain, how the pollen grains are forming? Center of the microsporangium, the tissues are known as sporogenous tissue, and these sporogenous tissue undergo meiosis, and these meiosis, there will be formation of tetrad, that means 4 cells, and these are haploid because these are gamete. So that means these are going to form gamete, so they are haploid, and this is known as pollen tetrad. So this process is known as microsporogenesis, means formation of microspores. So pollen grain means microspore, so here is the formation of pollen grain, so this is microspore. And later on, all these 4 microspores, they get detached from each other, and individually they are known as pollen grain. Individually they are known as pollen grains. That means after one meiosis, there is a production of how many pollen grains? 4 pollen grains. After one meiosis, 4 pollen grains. So this is the structure of pollen grain. Now I will show you the microsporogenesis once again in another picture. So this is the process of microsporogenesis. Pollen mother cell, in short we can say PMC. So this pollen mother cell, it is diploid, 2N. Then it undergo meiosis process, meiosis 1, meiosis 2, formation of 4 cells, 4 haploid cells, and these 4 haploid cells known as microspore, and they separate from each other and formation of pollen grains. This is haploid. Pollen mother cells diploid. So diploid change into haploid. The process is called microsporogenesis. Now the next structure, that is about, I think it is clear to all of you. What is the next? Next is structure of pollen grain. So the structure of pollen grain, once again, it is not so hard. It is the structure, only I have to explain the structure here. So the outermost wall of this pollen grain, it is known as exine. This is exine. Pollen grain is a round, near about spherical structure. Outermost is exine and it is made up of sporopollen. Then inner layer is known as intine and it is soft, elastic in nature. You can see here germ pore, a small gap where there is no exine, only intine present, that is called germ pore. Pollen, you can see 2 nucleus. One is called generative nucleus, another is called pollen tube nucleus. So this is the structure of pollen. Now you can see here the explanation of this structure. Once again, I will show you. That is exine. It is the outermost layer and it is hard layer. I told you it is hard layer. This is again another question of your board. That is sporopollen. Exine is made up of sporopollen, which is extremely resistant and can withstand high temperature, acidic and alkaline conditions and enzymes. So this is non-biodegradable. Okay, so it is exine is made up of sporopollen. And the next is intine, that is the inner layer, and it is made up of cellulose and pectin. It is a soft elastic layer. Okay, that means the pollen tubes are formed by this intine layer. The pollen tubes are moving out through this germ pore and it is formed by this intine. Now mature pollen grains. After maturation of this, the pollen grain, it contains 2 cells. The pollen grains contain 2 cells. So what are these 2 cells? One is called vegetative cell and another is called generative cell. So this is vegetative cell. Which one is larger? That is vegetative cell. So here it is written large with irregular nucleus, that is vegetative cell. And second one is generative cell, that is the smaller cell. So generative cell is small and floats in the cytoplasm. Altogether, both the cells, they comes in present in the same structure. It is not the 2 cells, it is separated. When pollen grain dehiscent from this, that means it is written here 60% of the angiosperm pollen grains are shed at 2 cell stage. 2 cell stage means that when pollen grain release out from this their plant's body, they are having they are of 2 cell stage. So it is generative and vegetative cell. Now this generative cell, it undergoes meiosis. Again this is important. Till I have not used the term gamete. Every time I am telling you that is pollen grain. Pollen grain is not gamete. Pollen grain means microspore. Another term we can use for pollen grain is male gametophyte. So generative cell, they divide and form 2 male gametes. 2 male gametes. So this is male gamete, formation of male gamete. That means male gamete where it is formed after division of this generative cell. Male gametes are formed. Right. So pollen grain, it is not male gamete. Pollen grain, it is male gametophyte. Male gametophyte. So pollen grains are male gametophyte. Remember this, right? Inside of this, you can see 2 male gametes. When generative cell, generative nucleus divides by mitosis, simple mitosis cell division, they forms male gamete. So this is about the structure of pollen grain, structure of, we can say, the microspore, and then male gametophyte. Here it is written the viability of pollen grains after they are shed depend upon temperature and humidity. It ranges from 30 minutes to few minutes. So these things are given there in your book. So this is today we have read about microsporogenesis and development of male gametophyte. Here's a question for you. What is the ploidy of vegetative nucleus formed by mitotic division of microspore nucleus? And second question is, what would happen if germ pores were absent in exile? So please check your understanding by answering these questions. If you are able to give answer, means you are getting the chapter. Here I have given link for video. So this is all about this topic, that is microsporogenesis, development of microspore or pollen grain, and structure of flower and structure of microsporin gel. So this is all about. Thank you.